Okay, so neurophysiology. So we learned a little bit about how general physiology, active potentials, we're going to start building upon these active potentials to see how we interpret things from the world and our environment. So what does the nervous system do? Obviously, a wide range of functions, unconscious control of visceral functions. So the fact that we don't have to think about breathing or our heart beating or digestion, it just happens automatically, helps a lot. It's composed of neurons and the support cells called glia. That's really all the brain and the nervous system is. It's just nerves and support cells. That's all it is. There's a ton of neurons there. Limitless potential for circuits and patterns of connectivity. Patterns, that's how we learn as humans. We are pattern recognition species. That's why repetition and constant studying helps, because you're looking for patterns on your own, and when you recognize the pattern, then you've learned the concept. That's why we have a lot of repetitive. With PA stuff, in the fall especially, we align everything. So when I teach patho, when I do cardio, we're going to do cardiopathophysiology, and at the same time, you're going to learn EKG, you're going to learn physical diagnosis for cardiology, you're going to learn pharmacology for cardiology, you're going to learn your CMS for cardiology. So everything goes at the same time. Repetition, you constantly get it a lot. So studying, you're looking for patterns. And when you realize the pattern, then you've made a connection and you'll learn what you're doing. So that's where our learning, that's why repeating things, writing things down, the writing, the actual act of writing the words helps you remember things. That's why we have you write things and stuff like that. Regional specialization. So what does that mean? We have the entire brain mapped out. We know what parts of the brain do what and what they're responsible for. So if you come in with a complaint, we can kind of figure out ahead of time what territory of the brain is responsible for these activities and why and what vessels may be involved that are causing these things to happen. So the brain is mapped out. We know what things do. So when you damage a certain part, we can figure out what the location is. Some anatomic terminology. Hopefully this is a review by now for you guys. Although, I don't think you had anatomy yet, have you? No. So tomorrow, right? So you'll get this tomorrow. So rostral and caudal, toward the head and the tail. So rostral is toward the head, caudal is toward the tail. An easy way to remember that too, the very end of our nervous system goes into a horsetail pattern. We call it cauda equini, because the cauda is the tail, equini, horse, resembles a horsetail. So cauda is always the tail end of our body. So above the midbrain, rostral is the front and caudal is the back. So anatomical position matters. When you're upright, above the midbrain, rostral is in the front, caudal is in the back. Okay, that's only above the midbrain. Below the midbrain, below the medulla oblongata, all that stuff, the brainstem, then caudal is the tail end, the back, and rostral is the front, or the head. Dorsal and ventral, okay? Dorsal is the back, ventral is the front. Think of a dorsal fin on a dolphin. It's on the back. Okay, so dorsal is the back, ventral is the front. But when you're above the midbrain, so we're referring to the brain itself, the dorsal is the top of the brain, the ventral is the bottom. Only when it comes to the brain, everything below the brain follows the normal pattern, but above the brain, it flips. Anterior and posterior toward the front or back of the body of the brain, respectively. So those are there too. When viewed dorsally, the nervous system is divided along the midline, so our medial sagittal plane, and has equal halves, so a right half and a left half of our nervous system. It should be the exact same nervous system on the right side as the left side. We should all be the same. When you put it in half, it should be identical to both sides. Medial and lateral, so medial is towards the midline, Lateral is away from the midline. So medial, lateral. Okay. Ipsilateral, 2 points on the same side. So we say someone has ipsilateral weakness. That means it's the same side as the injury. If they say that we have contralateral weakness, it's the opposite side. So contralateral is the opposite. Ipsilateral is the same. Again, just more definition words to start getting in your brain so that when I say ipsilateral, you know exactly what I'm talking about. We can also have different orientations. We can have coronal slices, vertically oriented at 90 degrees to the sagittal plane, and we have horizontal. I have a picture for you guys. Horizontal plane, perpendicular to the sagittal and the coronal. So here is the way to look at it, all the different words put all together. You can study this on your own. Again, whatever way works for you to remember these things, go right ahead. Doesn't matter how silly it is or whatever. So we have our neuron. We have the soma, which is the cell body, which is right there. We have neurites, thin cellular processes extending from the soma. The dendrite, or the axon, is what carries it the whole way, and the dendrites is what receives impulses from other neurons. So dendrites are over here. You can even call these neurites back there as well. But this is the direction of all the flow. Starts in the cell body and goes out towards the neurites. The dendrites receive the information from either another nerve, remember that signal cell communication, and that's where that happens. Some definitions for you: gray matter. Gray matter, whenever we refer to gray matter, it's always the cell bodies. So if you ever look at a CT scan in the head, you see gray matter is this, white matter is this. Gray matter is always the cell bodies. Yes, your question? No, I think neurites, consider them more at the very end. Kind of sends the signal to the next one. Dendrites kind of receive. So gray matter is your cell bodies. The cortex is the outer layer of the cerebrum. We have nuclei, loci. Ganglia is another one. These are collections of neurons in the peripheral nervous system. What else I want you to know? White matter. White matter is the axons. The axons. What do the axons have wrapped all over them that help with conduction? Myelin. Myelin is made up of what substance? It's fat. It's fat. Okay? What color is fat? Like you have like a steak or something when you don't cook it? It's white. That's why your white matter is all the axons, because it's all the fat. The cell body, the soma, does not have myelin. That's why it's gray. And the cell bodies are always gray. The axons will be white because of the fat content that wraps around the myelin, the tract of the neuron. A tract, so when you talk about a tract, is a collection of axons. So we have different tracts that go from the top through our brain down to our body that provide different types of movement, but they're all in a tract. It's just a collection of wires. Commissure, one side of the brain to the other. So the commissure is something that connects one side of the brain to the other. Okay, and obviously a nerve, bundle of axons in the PNS, except for the optic nerve. Optic nerve comes out of the brain, and they stimulated brain activity, like they take brain cells and let the brain develop and grow, just brain cells. And then they go long enough, it actually made optic nerves and started making eyes. Because the optic nerve comes out of the brain, not the brainstem. So we can divide our nervous system depending on where the location is. We have our central and our peripheral. Central, brain and spinal cord. Everything outside of that is your peripheral nervous system. All your nerves that branch off is peripheral. But your brain and the spinal cord are the central. They're covered in 3 layers called meninges. The outer layer is the dura mater. The middle layer is the arachnoid mater. And in this arachnoid layer, this is where you create and make CSF, the cerebrospinal fluid. And you have the pia mater, which is the innermost, highly vascular. When you talk about meningitis, it's an inflammation of these meninges that cover the entire central nervous system. So all that inflammation causes compression on the spinal nerve and the brain and causes the pain that you have with meningitis. Your peripheral nervous system, again, neurons and glia outside the meninges. So everything outside the meninges is peripheral. If it's inside, it's central. Our peripheral nervous system includes spinal nerves, cranial nerves, and our sensory receptors. So how do we receive the outside environment are all receptors. How our body moves through our spinal nerves and cranial nerves allows with all the movements with our head, a lot of that stuff. I talked about this already with meningitis. Encephalitis is actually the swelling of the actual tissue of the brain. It's the parenchyma. When you ever see the word parenchyma, it refers to the actual tissue of that organ. So in encephalitis, it's the actual swelling of the brain tissue itself. Meningitis is just swelling of the layers that surround it. We can also divide the nervous system into its functions. What is it doing? And we have two different types. We have autonomic nervous system, and we have our somatic nervous system. What does soma refer to? Body. So it's all your voluntary muscle contractions. So somatic nervous system, sensory and motor communication between the sensory nervous system, skin, skeletal muscle, and joints. Anything with the body receiving information is your somatic. Your autonomic nervous system is usually your visceral functions, digestion, blood flow, temperature regulation, reproduction. Those are the main functions of our autonomic nervous system. It's autonomic. We don't have to do anything about it. It does it automatically. Autonomic nervous system is broken down into three different parts. We have our sympathetic nervous system. This is your fight or flight. Okay, it's excitatory. Okay, if you're getting attacked by a bear, you want your sympathetic system to activate so you can do whatever it is you have to do. I guess what is that new thing, that 100 guys versus a gorilla? What they don't tell you is the minute the first guy gets mutilated, the other 99 will be running away because they're not going to stick around and fight when some guy's getting ripped apart. So that's fight or flight. What do you have to do to run away from something? Heart rate has to go up. Breathing has to go up. All to allow you to do an activity to run away from not being killed. So that's fight or flight. Parasympathetic is antagonist to your sympathetic nervous system, and just think of rest and digest. So right now, you all are sitting here, you had lunch, your parasympathetic system is in control right now. Okay. The enteric nervous system, this is that gut brain. So we'll get to that more when we deal with GI. But there is some sort of connection between the gut and the brain. One of the biggest neurotransmitters, anyone know what the neurotransmitter is involved between those two? Serotonin. Okay, serotonin is the one. And that's the link, they think, between like what you eat and your GI health and depression. Is the food that you're eating is not giving you the serotonin that you need in your gut to tell your brain, hey, we're happy. So a lot of it comes through the food you eat. So that's the enteric nervous system. Structures of the brain, we have the cerebrum, which are two cerebral hemispheres. Okay, it receives input and controls movement of the contralateral side of the body. So what does that mean? So, people who have strokes, if their right side is down, their motor system in the right arm is down, where was the lesion? Left side of the brain, because the left side controls the contralateral side. Like I said before, with neuro, if you come in with right arm weakness, I know exactly what side of the brain is affected and what blood supply was probably altered, causing this problem to happen. We have the cerebellum, which is behind the cerebrum. It's that little piece right in the back, cerebellum. Key part of our motor system, and big thing is coordination. Okay? What substance affects your cerebellum? Alcohol. So what tests do they do to see if you have alcohol intoxication? Field sobriety tests, which are what? Coordinated movements. In order to do coordinated movements, you need an intact cerebellum to do it. So a lot of motor problems, people have problems walking, initiating movement. It's problem with the cerebellum, and alcohol, we know, is toxic to the cerebellum. So if you're drinking too much and you're drunk, your cerebellar activity is diminished, and therefore you have problems with coordinated movements because of the alcohol. And then the last piece is our brainstem, and vital body functions is in the brainstem. If you're seeing a patient who had a stroke and they cannot support their heart rate or their breathing, that means the stroke hit the brainstem, and those are very, very, very bad prognoses. You don't want strokes in the brainstem because then you can't support yourself, you can't support your heart rate, you can't support your breathing, you need to be on life support because of a brainstem infarction or a stroke of the brainstem. So those are the major structures of the brain. We have our spinal cord as well. One of the main things I want you to know is that we have a dorsal root and we have a ventral root. Both of these, and I'll put them in different colors. Here's our dorsal. Here is the ventral, and they come together to make a mixed nerve or a spinal nerve, but they come together. Always, so always, write this down. Dorsal is always going to be sensory. The dorsal root will always take in sensory information from the outside world to transmit up to the brain. The ventral root is always going to be motor. The ventral root is motor. And that's always going to be that way. Then they mix together to make a spinal nerve, which then causes your dermatomes. So you guys anatomy learn about dermatomes yet? Okay. This is where a dermatome forms. It's sensory and motor, and they go to a certain location when they break off the spinal cord. So where they go, that is the dermatome. What else do I want to tell you here? So the CSF fluid, okay, AA, and you guys are doing your epidurals, okay? It's called an epidural for what reason? Where are you injecting it? Okay, that's the location we have to inject things because it's going to get to the brain and cause, pain relief or whatever you're doing. So your locations, and when you start doing your spinal nerve and taps and all that stuff, you have to know these meninges so you know which you're in the right layer to apply the medicine. Our four brain structures, obviously the cerebrum is the biggest part of our brain. Thoughts, perception, voluntary actions all come from the cerebrum. Okay, the frontal cortex, is not really developed until your mid-20s because that's where all your personality, judgment is all formed in the frontal lobe. Cortex is interchangeable with cerebrum, but refers to the surface of gray matter. So anytime you see the word cortex, it's the gray matter, and the gray matter is referring to what? Cell bodies is the gray matter. Okay. The surface area of our brain is increased through folds and fissures. So we have, instead of having a smooth brain like a koala or a panda bear, because they are really stupid. No, you can put a plate of like eucalyptus leaves for a panda to eat, which they eat all the time off the tree. But because it's on a plate, they don't know that it's eucalyptus, and they won't eat it because they're really stupid. Because they have a smooth brain. We have folds. We have sulci and gyri, which increase the surface area. By increasing the surface area means we have more neurons, more cell bodies, more connections, the smarter you are. So we have these large sulci that divide the cortex into a frontal cortex, a parietal, occipital, temporal. They have an insular lobe, but it's very hard to see. You got to pull some layers back. But these large sulci make the different lobes of the brain. So this is Einstein's brain. Look at all those folds that he has compared to a normal human brain. Okay, these extra folds right here, that's your temporal lobe. That temporal lobe is a lot of spatial stuff, thought. That's where the temporal lobe has all that stuff. And comprehension. Temporal lobe, again. Supposedly his brain went missing. I don't know where it is, but I don't know about that. Other forebrain structures, the lower part of the forebrain, we have our thalamus and the hypothalamus. The thalamus, relay station for sensory pathways. All the sensory pathways so for you to like feel your pen that you're writing with, the sensation of your phone vibrating in your pocket, all of that has to go through the thalamus before it goes to the cortex. So the thalamus is a sensory relay. Sensory pathways go through the thalamus to the cerebral cortex on the same side, but convey information from the opposite side. So again, the left side of the brain controls the sensory input for the right side of the body. So if I can't feel anything with my right hand, I can't feel what I'm doing, my left brain is having a problem, because the left brain controls the sensory for the right. Cortical white matter, again, it's myelinated axons. That's why it's white, because of all the fat on the myelin. The corpus callosum is the link between the two hemispheres. Because normally we have two separate sides. We do have communication between them. That's where the corpus callosum comes in. It allows communication from left side to right side of the brain or whatever. And we have the gray matter, deep cerebral nuclei, or the basal ganglia. And this is where other higher motor and higher thought process functions occur in these areas as well. Forebrain structure, again, the hypothalamus is ventral to the thalamus. It's hypothalamus. It's below the thalamus. That's why it's called the hypothalamus. It's the command center for the autonomic nervous system. So the hypothalamus is very, very important. And also all the endocrine stuff via the pituitary gland. The hypothalamus is also involved with a lot of our endocrine control. Other sections of the forebrain, amygdala and the hippocampus, and that's our emotion and memory. That's where they're stored. Our retina and optic nerves are in the forebrain, and the olfactory bulb. So vision and smelling are in these areas as well. Let me read that one. Okay. Finally, people got it. Okay. Midbrain. So now we're moving into the brainstem, getting out of the actual brain itself. Narrow portion of the brainstem, conduit for nerve tracts between the forebrain and the hindbrain. Again, it's just things go through it. You don't need to go crazy here knowing all these structures in here, so don't worry about the different structures inside. Substantia nigra. I do want to mention this. So this substantia nigra is right there. Nigra because it's black. Why is it black? Anyone know? What neurotransmitter is in this area in very, very, very high quantities? Anyone know? Dopamine. This is dopamine. Dopamine makes it black. When you don't have any dopamine, this thing turns white or turns gray. And what disease do you get with lack of dopamine? Parkinson's disease. And we'll learn later on how Parkinson's or dopamine aids in your movement. And while when you don't have the dopamine, you have problems with movement. We'll get to that later on. Okay. Your hindbrain structures, here's your cerebellum, the pons, the medulla, as you keep going down the brainstem. We already talked about this with different sides and what they do. Hindbrain, brainstem, just a conduit for many nerves. And obviously we have our midbrain, pons, and medulla. And these are associated with a lot of our cranial nerves because they come out of the brainstem as well. As far as your cranial nerves, I'm not going to quiz you guys what cranial nerve does this. We're going to go through them in some things as we go. But again, I know you guys have anatomy. You'll have to know this anyway. So again, like these stupid mnemonics, cranial nerve 1, you have one nose, you have two eyes. There's an easy way to remember the first two. One nose, two eyes. And the rest of them figure out on your own. But whatever you use to remember. But they're there. So these are all different nerves. Oculomotor, that's a lot of it has to do with your movement of your eye. The iris, constriction, dilation is all cranial nerve 3. Cranial nerve 4 is your trochlear nerve. different proprioception, where you are. A lot of it has to do with eye movement. Trigeminal nerve, we have 3 different divisions, trigeminal, 3 of them. One is for your eye, one is the upper jaw, one's the lower jaw. Just remember, we got 3 divisions in the face, and they help control your facial movements. Your abducens is 5, is for eye movement. Your facial nerve, obviously, one of the things you guys will do in PA school is you're going to learn how to test for each of these cranial nerves. So you'll know which one you have to do. You guys haven't started it yet, but you will. What you have to do for each nerve. Vestibulocochlear, we're going to focus a lot, probably next week when we get to like how we hear, how our balance works. Glossopharyngeal, the tongue and taste. Vagus nerve is a big one. Vagus, it's called vagus for the word vagabond. What does that mean? Vagabond is someone who wanders. It's a wanderer. The vagus nerve is one of the longest cranial nerves. It starts in the brain and actually goes all the way down into your abdomen. And it innervates down there. Even to your diaphragm is the vagus nerve. So it's a wanderer, innervates a lot of different things. Accessory nerve helps with breathing and your soft palate. Hypoglossal as well. So again, they're there. You'll learn them as you go. Structures of our peripheral nervous system now. So now we're moving on to the periphery. We have 31 pairs of spinal nerves. 12 pairs of cranial nerves. So don't confuse those, the cranial nerve versus our peripheral nerve or spinal nerve. The optic nerve is considered part of the CNS because it's located within the meninges, like I mentioned before, like the brain, the eyes come out of the brain. They don't come out of the spinal cord, the optic nerve. So that's why it's part of the central nervous system. And again, except the vagus nerve, everything else stays within the head and neck region. But that vagus nerve, it's a wanderer, so it leaves the neck and it goes into the thorax, into the abdomen. Okay, that's it as far as the structures of the brain are concerned. Now let's move on to our brain extracellular fluids. Our brain is limited by the bony skull. So what does that mean? Our skull, when you're first born, all those little fusions in the skull haven't fused yet, so our brain is malleable. So if you've seen a newborn baby, some of them have a cone head from getting out, but then it goes back to normal and they're okay. As you get older, all of those sutures fuse and your skull becomes a fixed size. So your brain can't get any bigger. So if you have brain swelling, there's nowhere for that swelling to go. If you bruise your arm's going to swell because your skin can get bigger. The brain swelling, there's nowhere for that stuff to go because the skull is fixed. So our volume of our brain is fixed. Your eyes, when you're born, your eyes are the size they are forever. That's why babies have like big eyes. Because they're like normal adult-sized eyes with a small little head. So they look huge because your eyes don't grow. Once you're born, that's the size of your eyes. So again, if volume increases, you get increased intracranial pressure, because all this pressure is building up inside the brain, there's nowhere for it to go. And when that happens, you get decreased brain perfusion. And that's when you start getting altered mental status, passing out, because the brain is not being perfused. So the composition of the brain extracellular fluid is very, very important to keep stable. We don't want that stuff to be mixed with the outside environment. We don't want infections to get into the brain. So we need to make this blood-brain barrier. So there's 3 components to our extracellular fluid in the brain. We have blood plasma, we have the interstitial fluid, which comes into contact with all the neural cells, and we have our CSF with the ventricular system, that cerebral spinal fluid. All that fluid provides cushioning and also a free exchange of solutes. So that's what composes the extracellular fluid of the brain. We have the blood plasma, interstitial fluid that kind of bathes the tissues, and we have the CSF within the ventricular system. The ventricular system is lined with choroid plexus epithelium. All you gotta know about that is that it makes CSF. That's it. It just makes and secretes CSF. The CSF flows over the brain, it goes down to our spinal cord, comes back up, it floats around, and it circulates. It drains back into the venous system via the arachnoid granulations. So the epithelium creates the CSF. The arachnoid granulations absorb the CSF, so we're always constantly making new CSF and replacing it. So we have a way to make it, and we have a way to get rid of it. So to maintain a stable CSF, the rate of production must equal the rate of elimination. And as long as that happens, you'll have no problems. If the absorption of the CSF is diminished, what's going to happen to the volume of CSF? It's going to increase. And it happens a lot. If you ever hear this term, the disease normal pressure hydrocephalus, that's too much CSF on the brain, and it causes some motor problems. You would think they had Parkinson's the way they present. If you did a scan, you'd see their ventricles are huge, and it's just due to overproduction of CSF or lack of removal. So we want that to be constant. We want our CSF volume to be the same. So the absorption and the creation have to match for that to happen. So what are the functions of CSF? Shock absorber, maintenance of constant internal environment in the central nervous system, and route of removal for metabolites in the brain. We have to get rid of these things. They can't just stay in there. We have to flush these pathogens out. So there's a hydrocephalus I just talked about. So we have a thing called the blood-brain barrier. We don't want our blood composition to mix with our brain. We want to keep it separate. So we have a blood-brain barrier for this purpose. We have capillary endothelial cells that enclose the brain capillaries and form the selective blood-brain barrier between. So you can see here the brain capillaries. These are the astrocytes. So the astrocyte is the glial cell that provides one of the barriers for the blood-brain barrier. So these little podocytes that you see that cover this entire capillary prevent metabolites from leaving the blood to go into the brain. Only certain molecules can get through. Obviously, which ones will go right through? What type of substances will go right into that blood-brain barrier? What are some things? They go right through. They don't have to worry about, there's no, oxygen would help, right? CO2, all these gases, they go right through real fast. So like a lot of the anesthetic drugs, they have to cross that blood-brain barrier. They have to be lipid soluble to get through. And that's why a lot of the anesthetic stuff is that built that way, so it can cross that blood-brain barrier to have an effect in the brain. Also, we have these neighboring endothelial cells that have these tight junctions that also prevent movement as well. So this is a typical systemic capillary. It's very leaky, but in the blood-brain barrier, they're not leaky. They have these podocytes that surround the capillaries, preventing metabolites getting into the blood-brain barrier. So these are the ones that can get through very easily. O2, CO2, nicotine, ethanol. Okay? Nicotine, you smoke a cigarette, you may feel euphoric or high afterwards for a few minutes because of the nicotine getting into the brain. Same thing with alcohol. It can get into the brain. Obviously, we know it goes to the cerebellum with the alcohol. Non-lipid soluble are unable to diffuse into the brain. The only one is glucose. Okay, our brain only works off of glucose, not sucrose, lactose, any of those things. They have to be converted to glucose in the body so that glucose can then get to the brain. That's what it works off of. And remember, we talked about those GLUT transporters. It just grabs the whole glucose molecule, brings it through the protein, and dumps it out inside the cell. That's how glucose can get through the blood-brain barrier, through a membrane-spanning protein. It brings the whole thing in. We do have some areas of the brain that are technically considered outside this blood-brain barrier. They're still in the brain, but we do have a certain, some areas that have a very, very lax blood-brain barrier to allow other things to get through. These are called circumventricular organs. So these organs can sense what's going on in the blood in the body. So some examples, posterior pituitary. Very important to know When we get to the posterior pituitary, what hormones come out of that have to do with volume status of the body. So we want, the brain needs to know what's our volume status and alter things based off of that. Subfornical organ, median eminence, pineal gland. This last one has a big long word, but I want you to remember just the OVLT. The OVLT is very important when it comes to body temperature. So if you've ever had a fever and chills, it's because the OVLT senses some cytokines that are floating around your body, and hey, we got to kill it off. So let's raise our body temperature to kill off all these bacteria in our body, or the viruses, or whatever it is. But the brain has to sense, hey, we have cytokines, which means there's an infection going on. The brain senses these cytokines, tells the hypothalamus, hey, we need a fever, raise it up, and then you get a fever generation. And we'll go through the fever generation in a few minutes. But again, exposed to solutes in the blood so it knows what's going on with the body. And you can skip that one. Okay, cell neuroscience. Okay, sophisticated higher function of the nervous system because our neurons can communicate. Do you ever hear the stoned ape theory about why we have communication, why we can talk? Apparently the apes, when they were like millions of years ago, were hunting. So the way they would hunt was to follow the dung of the animals that would travel. Well, what grows in feces? Mushrooms. So they were eating the mushrooms and they were tripping balls. And that's how they had to figure out a way to communicate what they were seeing, because they were seeing colors, all this weird stuff. And that's where like language came from, was tripping off mushrooms. And that's why we can talk. And now we got to pay taxes because of that. So that's what happened to us. That's how we can do all that. One neuron can reach thousands of inputs from other neurons. And so what happens is we get produce output signal via an axon. The size, shape, and function of our neurons change when we classify them. So let's talk about different types of neurons or different neurites. We have a unipolar, which is a single neuron, which is most of our sensory neurons are going to be unipolar. You know, one cell body, one direction is where it goes. You can have bipolar, which has two neurites, so it receives information from two different areas, going to the cell body, so it's two different areas. And then finally, we have this multipolar, which is the most common type of neuron, which all these little dendrites at the end are receiving input from other neighboring neurons. They're not in the picture, but they're right next to it, and they're going to communicate to cause all these communication to occur. So the way you organize these dendrites in the cerebral cortex, there's two patterns. There's pyramidal neurons and there are stellate neurons. This is the way they're shaped. Pyramidal is in a pyramid track, stellate is star-shaped. Pyramidal neurons are all your motor neurons. They go in that pyramidal tract. So I mentioned yesterday, I forgot where I mentioned it, but watching those, the drug commercials for all the side effects and like listening to what you're hearing. One of the things you may hear with certain drugs, they cause what are called extrapyramidal symptoms, or EPS. Certain drugs cause motor problems. They're called extrapyramidal because they affect the motor system, not with the pyramid. They use outside the pyramids to affect your motor system. So if you ever hear of extrapyramidal side effects, it's meaning they're following motor systems that are outside the pyramids. And then stellate, you're just talking about star-shaped neurons. We also talk about the axon length. How long is the axon? Golgi type 1 are very long, and they go from one region of the nervous system to another. So Golgi type 1 are the big ones. And then we have Golgi type 2. These are very short axons that are usually just found within local circuits. They don't really leave that little area that they're in. So Golgi type 1 will go from system to system. Type 2 will stay within the system. We can also classify our neurons based off of their function. What are they doing for us? So a primary sensory neuron, body surface, and they convey sensory info to our central nervous system. We have motor neurons, and they end on muscles, and they initiate muscle contraction. And then we also have one small type called interneurons. These are usually within reflex arcs. So you touch a hot stove, you pull away real fast. That is a reflex arc. Usually there's an interneuron there between the sensory neuron of you feeling the heat, and there has to be a motor neuron for you to pull your arm away. So there's two nerves that are involved with that reflex arc. The sensory coming in of the temperature, heat, and then the motor action of you moving away. An interneuron is in between those two neurons to connect them. That's all the interneuron is. We can also dictate the neurons based off of what type of neurotransmitter they release at the end of the nerve. So if it releases acetylcholine, it's considered a cholinergic neuron. What have we learned so far? What type of nerve is a cholinergic neuron? Motor, right? All motor nerves release acetylcholine as the neurotransmitter. So all motor nerves are also known as cholinergic neurons because they release acetylcholine at the very, very end. So we have our axons and presynaptic terminals. So when we talk about neurons, they're all going to have the same parts. The body, the axon, and the neurites at the very, very end, or the terminal boutons at the very end of that neuron. At the end here, it's either going to go to another nerve, right? Going to send the signal to another nerve, or it can go to a muscle and tell a muscle what to do. So something's going to be at the very end of that terminal bouton, whether it's a nerve, a muscle, something's going to be there. So the initial segment, or the spike initiation zone, is just beyond the axon hillock. So right here is what we're talking about, this initial segment. So all of these are receiving inputs from other neurons. The impulses all collect in the cell body before it travels down the neuron. This area that I circled, this initial segment, is a very important area when it comes to inhibition. If I want to inhibit this nerve, I inhibit it right there. Because any signal that comes in from all those different dendrites around it all have to funnel through this one area. So if I want to inhibit a nerve, I hit that area and I shut down any impulse that can get out. So that's why that area is important, that initial segment. No myelin, high density of voltage-gated sodium channels. What is that going to do when they open up? What's it going to start? Action potential. And that action potential is going to go all the way down. It picks up more speed with these nodes of Ranvier, right? The propagation of the impulse. And then we come down to the terminal boutons where vesicles contained quanta. What type, okay, let's start thinking. What type of voltage-gated channels do you think would be very highly concentrated down there by the green? Calcium. Why do you think calcium would be very high in this area? It's going to have to cause your exocytosis. So it makes sense that all the calcium channels would be right there. So as this impulse comes down, we only have really like your sodium channels up top through the whole thing, getting that action potential down all the way. But when it gets down to the terminal end, this is where your calcium channels are, because when calcium comes in, it's going to provide the stimulus for the exocytosis of all the neurotransmitters. So that's why the calcium receptors are always down there. That's where they're needed. They need it for exocytosis. Axoplasmic transport, okay? Stuff has to move up and through this nerve. So if you cut a neuron, the distal part degenerates in a process called Wallerian degeneration, which is why it's very hard. If you cut a nerve, it's very hard for it to grow back. Because when that nerve cuts, this Wallerian degeneration wants the nerve to retract and get smaller and pull away. We also have a transport of materials through the nerve. The nerve is basically like a big train track, and these little proteins sit on top, and they can travel down the protein. We have microtubules, and they run longitudinally like a train track. Microfilaments, there are actin molecules, and we have neurofilaments. These are the long protein ones. The direction of the movement also matters. We can have anterograde transport from the soma to the axon terminal. Retrograde transport, which is axon terminal to the cell body. So we're pulling in and we're bringing it up into the cell body. It's not going out to the dendrites. And we also have these little motor proteins, which are like the boxcars on the train tracks, which help carry the material back and forth. This retrograde transport is why herpes never goes away, because the herpes simplex virus goes into the nerves, goes into the terminal endings, and through retrograde movement, the virus works its way back into the cell body. And then it sits there and waits. And then stimulus, stressful, whatever, the virus pops out through anterograde motion, now goes out to the end, and now you have a canker sore or whatever. Herpes lives within the nerves. You can't treat nerve stuff. There's nothing to get into the nerves. That's why you can't treat herpes. It lives forever. Because it lives in the nerves. Nerves have no blood supply. What medicine can I give that's going to get into the nerve? So we don't have it, so they live forever. So I mentioned this already, the herpes. Neuroglia. So these are not the neural cells. Now these are the supporting cells, the glial cells. A lot of the brain tumors are glial cells, like glioblastoma. It's referring to the glial cells that's causing the cancer. It's not the nerve, it's the surrounding cells. There's only a few cells we got to know. Astrocytes is scaffolding, so it provides some sort of support for the different nerves. Has glucose. The other big thing, part of that blood-brain barrier. So the astrocytes provide the blood-brain barrier. They provide that podocyte that lines this capillary, preventing molecules to get through. The next one, microglia, immunity. That's all you gotta know. Microglia is the immunity part of the brain. Oligodendrocytes. They produce and maintain myelin in the CNS. Anyone know what cell makes myelin in the peripheral nervous system? Schwann cells. So Schwann cells and oligodendrocytes are the same thing. They produce myelin. The only difference is where they're located. Oligodendrocytes are for CNS myelin. Schwann cells are for peripheral nervous system myelin creation. So let's talk about synaptic transmission. Presynaptic axon terminals store that neurotransmitter. The action potential from the axon depolarizes those axon terminals. This is that wave of depolarization caused by what ion movement? Sodium. And that nerve impulse travels the length of the axon. Depolarization at the end causes calcium to go into that axon terminal, the terminal boutons or whatever. Then exocytosis occurs. Neurotransmitter interacts with a specific receptor on the postsynaptic side. And then the last thing is, how do we stop the communication? We started it with the depolarization, the calcium exocytosis. We have to stop the communication now. So we have to get rid of this neurotransmitter that's in that little cleft. We have to get rid of it. The ways we get rid of it, either it diffused backed up back up into the presynaptic, repackaged, ready to go again. Enzymatic degradation. What enzyme is responsible for degrading acetylcholine? Acetylcholinesterase. That's the enzyme that's going to break down. Or we can reuptake it and redo it, use it again. Those are the ways you get rid of it. So this is a way of looking at it again. We have our active potential coming down the nerve, down the axon. We come down to the terminal button or the N-terminal. This is where our calcium channels are, the voltage-gated calcium channels. So when the voltage comes down finally to the terminal, it will activate the calcium channels, bringing calcium in, and we get exocytosis to occur. Here's that neurotransmitter being released, and then it's going to bind to the postsynaptic membrane at a specific receptor for that neurotransmitter. Then the cell have a response. Is it going to change the membrane potential for the next neuron to cause an action? Will it cause some biochemical cascades to occur? Will other hormones get released? Or regulation of gene expression? When we get to hormones like thyroid and steroids, they get into the DNA and cause changes, but we'll get to that later on. So now let's go over some neurotransmitters. Acetylcholine is the first one. Do you need to memorize all that? No. Some people like the knowledge behind. I will say throughout the semester sometimes it magically happens. And when I say it magically happens, it just means that you don't need to understand the process. You just need to know that it ends there. So when I say that, I'm not saying you don't need to know what that is. It's just that it's not really important knowing the process, but knowing where it ends is important. So acetylcholine, how is it synthesized? Acetyl-CoA and choline acetyltransferase is the enzyme that synthesizes acetylcholine. You have to know the metabolites. What is metabolites, or how does it metabolize? Acetylcholinesterase metabolizes acetylcholine. Okay, uses, somatic transmission. That's our motor system. So we know acetylcholine is involved in the motor side. It's also involved with autonomic transmission as well. So acetylcholine will pop up with your autonomic nervous system as well. Receptors. You have to know this one too. There are only two types of receptors for acetylcholine. We have nicotinic and muscarinic. Where are nicotinic found? Skeletal muscle. Muscarinic we'll learn as we go. But for now, acetylcholine, made by choline acetyltransferase, metabolized by acetylcholine esterase, uses, somatic, autonomic, receptors, muscarinic, nicotinic. All you got to know. Remember, definition, learn that they go together so that when we talk about these things later on, more detail, you'll understand what we're talking about. So that's acetylcholine. The next one is norepinephrine. This is also known as noradrenaline, same thing. But norepinephrine is a neurotransmitter. Synthesis made from tyrosine in the diet. Aged meats and cheeses. Later on, you get to some diseases that some people, when they eat these types of foods, they get really bad headaches. If you have migraines, you may have been told avoid chocolates and wines and cheeses because they have high amounts of tyrosine, which can cause your norepinephrine to go up and cause problems with that. So that's why sometimes you'll see about foods. Metabolism, just know COMT and MAO. Now, MAO, monoamine oxidase, is how you metabolize norepinephrine. There are classes of drugs we give for depression and anxiety, and they're called MAOIs, which means it's a monoamine oxidase inhibitor. It's going to inhibit the monoamine oxidase. So, like, for example, your test is 50 questions. Most of them are just straightforward. What is this? What is this? What is this? I do throw a few thinking questions in there to get your mind thinking about where we need to go. One of the thinking questions is this. If I gave you an monoamine oxidase inhibitor, which limited the amount of MAO you had available, what neurotransmitter would go up in concentration? Norepinephrine. Why? Because I'm eliminating the monoamine oxidase. Nothing is going to metabolize the norepinephrine. It's going to stick around longer, and it's going to build up. So COMT, MAOs are how we metabolize and shut off norepinephrine. Function, secreted by brainstem, hypothalamus, helps with wakefulness, generally excitatory, so it's noradrenaline, it's one of those adrenalines, it's going to stimulate you to do something. Receptors. When we're dealing with norepinephrine, we call them adrenergic neurons because of the adrenal gland, which is where a lot of our norepinephrine comes from the adrenal gland. So any type, whenever you see the word adrenergic, think norepinephrine, also think sympathetic nervous system. Okay, norepinephrine is going to stimulate your sympathetic nervous system. The receptors for norepinephrine are your alphas and your betas. So just start, you don't have to know what they do, just associate. Alphas and betas, adrenergic neurons. Cholinergic neurons, what do they secrete? Acetylcholine. Adrenergic, norepinephrine, epi. They both will bind to alpha 1 and 2 and beta 1, 2, and 3. Don't worry about the numbers right now. You'll get that a lot later on. But keep that in your brain and keep it in your mind. Norepinephrine, alphas, and betas. That's what they stimulate to. Here's our dopamine. Again, tyrosine, meats and cheeses in the diet. It's for dopamine. Metabolize, same, COMT and MAO, which is why we get the MAOIs, which helps with dopamine, because dopamine is a happy hormone. It makes you happy. That's why people have that dopamine, like they call it dopamine withdrawal, try and get like excited or something. That's why drug abusers, they want the dopamine. So what happens is they need more, stronger doses to get the same dopamine amount, and that's where you get this tolerance to drugs and stuff like that. Why people start going stronger doses, because they want the dopamine hit. Function, there's a couple of different functions with regards to dopamine. Nigrostriatal, okay, it's black stripes. Black stripes, dopamine, substantia nigra, movement. So dopamine does have a role in our movement. The mesocortical pathway is our reward pathway. Okay, so when you guys got a great test and you get a great score on your first exam, your dopamine is going to rise because you feel great because of the reward pathway. That's where the drug abusers come in. People were the addiction because they want the dopamine hit. They don't get it anymore. And the last one is this tuberoinfundibular pathway. Don't worry about the big word about it. Just it goes to the pituitary. Don't go crazy. But the first two you should know. Movement, reward pathway for dopamine. Very, very important. Receptors for dopamine. There are only two, D1 and D2. So dopamine 1, dopamine 2. So those are the two receptors for dopamine when dopamine is the neurotransmitter. Here's our serotonin, made from tryptophan. Okay, again, in our food, turkey is a big one. Metabolized, MAO. Again, MAOIs help with serotonin concentration. Concentration of serotonin and dopamine go up, you feel happy. Function, altered mood, role in anxiety, nausea and vomiting. We mentioned before a little bit about serotonin also coming from the gut. So there's some involvement there. Cerebral blood flow. Anyone here have migraines? Okay, what do you take for your migraine?
I just do Imitrex for...
Okay, so Imitrex is a 5-HT agonist. So the word agonist means what? Is it going to stimulate that receptor or inhibit the receptor? Stimulate it. Serotonin helps with blood flow to the brain. That's what causes migraines. Actually, it's dilation of the vessels what causes migraines. The vessels get big, and it causes a headache. That's why caffeine helps. Caffeine is a vasoconstrictor. You take caffeine, it helps with your headaches because of the vasoconstriction. Serotonin also has a role in migraines as well. So that's why we give serotonin agonists to people with migraines to help with the cerebral blood flow. Receptors, there's a whole bunch of them. Don't worry about them right now. They'll tell you which one it actually is. Yeah, so serotonin and dopamine are technically the only two things you actually enjoy. There's chemicals that give you a feeling, and that's all you are trying to get. GABA, gamma aminobutyric acid. It's made from glutamate, also requires some vitamin B6. Metabolized by GABA transaminase, so know how it's metabolized. The function, generally inhibitory. So it's going to inhibit. GABA is inhibitory. Found throughout the CNS, it's the most common inhibitory neurotransmitter we have in the brain. So, look what it does. We have two GABA receptors. GABA A opens chloride channels. We talked about this a little bit yesterday, but now that we've actually gone through an action potential, hopefully it makes more sense now. What is our normal resting membrane potential? Okay, what is our normal threshold? Okay, so right now, in a normal healthy person, there's 15 units, 15 millivolts difference between our resting and we get the threshold. So it's pretty easy. GABA opens up the chloride channels. What happens when chloride goes into the cell now? What charge is chloride? Negative. So all this negative ion is going into the cell now. So what do you think is going to happen to that resting membrane potential with all this negative chloride going in? It's going to go down, right? Become more negative. So now, when we add GABA, we may go to negative 90 millivolts as your resting membrane potential. Now, you're 25 units away from threshold. It is harder to get to threshold. That is why GABA is inhibitory, because it opens the chloride channels, making that membrane potential more negative, making it harder to get to threshold, thereby inhibiting the brain. So that's what GABA does. Glutamate is the stimulatory one. What's MSG? Monosodium glutamate. Okay, it's monosodium glutamate. So for people that say years ago, no MSG because it caused headaches. It doesn't really cause headaches. It just made your pressure go up a little bit and made you more stimulatory in your brain, more active because of the glutamate. And it has some people that get weird side effects from that. They didn't like it, so they got rid of it. But monosodium glutamate is basically like salt, like with the flavor ramped up a lot more. It's like, anyway, so flavor ramped up, just a flavor additive. Metabolized by glutamic acid decarboxylase. Function, primary excitatory. It's the main excitatory neurotransmitter in the brain is glutamate. Receptors, NMDA, AMPA. I want you to remember the NMDA one, because one of the drugs that we give, and when you start learning about drugs, they're not very innovative with the name of the drugs, because the name of the drug to block the NMDA receptor is called Namenda. That's all. Namenda, and it took the receptor and made a word out of it. So Namenda is a drug that's going to affect the NMDA receptors. So there's no NMDA receptors. It's for glutamate. There are other neurotransmitters as well. Glycine is one of them. Histamine is a big one. Neurons in the hypothalamus. Histamine, effect on arousal, body temp, vascular dynamics. Why do you take what does histamine do? Anyone know what histamine is? Inflammation. It makes your capillaries leaky. So when you have an allergy attack, you start dripping from your nose like crazy. So what kind of medicine do you take to prevent that? Antihistamine, which blocks the histamine, which, when you block the histamine, arousal, you get tired, right? You ever take Benadryl and get knocked out? It's because it knocks out your arousal. Antihistamine, that's what they do. This last one right here, these H1 receptors, that is dealing with another way to prevent acid reflux. What did we learn about yesterday with acid reflux? What proton? Well, I mentioned it, the proton pump inhibitor, right? You block the hydrogen from being released. Another way are these H1 receptors. They're called H2 blockers. Have you ever heard of a GI med called, it's referred to as an H2 blocker? You're blocking histamine because histamine by itself will cause more gastric acid production. So by blocking it, you reduce the gastric acid and help it that way. Nitric oxide, the big thing with nitric oxide I want you to know, vasodilation. Nitric oxide is very, very potent vasodilator. Anyone work in the ER as a scribe? What do they give for chest pain patients? Nitroglycerin, right? Which is nitric oxide. Why? Vasodilator properties. If you're having a heart attack, that means one of your vessels is constricted or blocked. So by giving a nitric oxide vasodilator, we make the vessel bigger. Hopefully blood can get through that area. Cannabinoids. Okay, so our marijuana, we have receptors for that as well. And obviously, this is listed not inclusive. There's tons of neurotransmitters, but these are the big ones that we'll be doing that you need to know. And this we already talked about already with regards to GABA and what it does with our CNS. Let's take a break. Come back at 3:30, and we'll finish up. Okay, so okay, guys, let's get going so we can get out of here. So we learned before when we talked about muscle recruitment, there's two different ways to do it. One is with temporal summation, one is with spatial summation. So the temporal one dealt with what? Time. How fast was the nerve being stimulated? Spatial refers to the multiple inputs coming in at once. Remember, the goal is to get to threshold. Because once you get to threshold, then everything, all the sodium channels open up and you have an action potential. So the goal is to get to threshold. You need a strong stimulus to get to threshold. So what we're trying to show here is how we can get different grades of stimulus through different means. So we have our resting neuron. Here is, negative 65, negative 70, whatever the number is, doesn't matter. If we wanted to have excitatory, we have excitatory neurons coming in, which provide enough of a stimulus to get you to negative 45, which is threshold, and then you have the stimulus going down the row. So that's excitatory. Sodium influx causing excitation. Here is the inhibited neuron. And the ways that we can inhibit a neuron, calcium influx, what's that going to do to the resting membrane potential? Make it more negative, decrease it. The other way is to cause potassium to leave the cell. Okay, remember, potassium is a huge positively charged ion. As it leaves the cell by itself, it's going to make it more and more negative. If it's more negative, it's harder to get the threshold. Therefore, it's inhibitory. So, when you're reading these, excitatory, sodium influx, that's how you're going to excite that neuron. You need it to depolarize. Sodium has to move. The way you can inhibit a neuron? Increased chloride influx. What neurotransmitter is going to cause chloride to come into the cell? We just learned about it. Which one is inhibitory? GABA. GABA is inhibitory. It opens the chloride channels, makes the memory potential more negative. Another way, this is talking about summation, millivolts, milliseconds, over time, what's going on. If you have 16 synapses firing at once, you're going to have enough of a stimulus to get above threshold. That's all we're showing here. The more stimuli you have, the stronger the stimulus becomes, which is hopefully going to be enough to get you above threshold. If you see here, 8 and 4 aren't strong enough stimuli to get you above threshold. So again, just showing you, the more inputs you have into a nerve, the stronger the stimulus, the stronger the ability to get above threshold to have that all or none happen. So you can have an active potential. So it's called summation. You're adding stimuli together to get above threshold. The two ways to do it, spatial, so you have multiple inputs from different neurons, or temporal, successive discharges of one nerve. Either way, you got to get the threshold. These are just two ways to do it. You add the forces together, the stimuli, and if it's strong enough to get you above threshold, you're going to have an active potential, you're going to have a movement. So that's what we're talking about here. The biggest area where we want to affect, remember there's this axon hillock right there, the big area we can cause some changes. So, effects of synaptic excitation. These are all the dendrites that are coming off the cell body. And if you see all these E's, look at the numbers where the E's are. The E is the excitatory neurons. They're all at threshold. Okay? Now, look where the inhibitory ones, and I know there's some here, but this is where the big ones are. The inhibitory neurons collect at the axon hillock before it goes off to the neuron, to the nerve, the axon. So the inhibitory ones right here, look at those millivolts. Look at the resting memory potential, negative 70, negative 75. It's making it more negative, so it's harder to get to threshold. That's all I want you to know from this one. Excitatory neurons are more peripherally at the neurites. The inhibitory neurons are going to attack the axon hillock because you don't, what if I just inhibit this neuron? What if I knock that out, but I still got 3 other neurons that are still receiving excitatory impulses? I'm only blocking one. I still got other ones that can still get through. But if I block it there, it doesn't matter what happens out there. It's going to get blocked before it gets out to the neuron. So by having the inhibitory neurons there, it's a safety mechanism so that none of those excitatory impulses can get through. Because you block it at the axon hillock, nothing can get through. And that's why a lot of the inhibitory neurons are going to be there because it can block any impulse getting through. So location matters for inhibitory ones. Some special characteristics of synaptic transmission. One is fatigue. They can't just keep going all the time. Eventually they're going to fatigue out. They're going to run out of neurotransmitter. It's not going to work anymore. They're going to fatigue out. So initial excitation discharges are rapid but slow and they die out. Important for termination of seizures. Okay, you have to stop a seizure. You can't let them go for a long period of time. Protective mechanism that prevents neuronal damage. If you keep exciting the neurons, you're going to fry them. And one of the new things we're doing now for like strokes is the area of the stroke that had the ischemic event is dead. It's not coming back. The problem is the area around it, the ischemic zone around the injury. That is under a lot of stress. A lot of glutamate is being released there. What does glutamate do? Stimulatory, excitatory. So you want to cut that glutamate. So some of the drugs that we give for strokes are glutamate antagonists to stop the glutamate from going crazy and frying the brain around the injury. So that's kind of where that comes in. So we want to prevent neuronal damage. And the mechanism by which it happens, loss of presynaptic vesicle content. Remember, every discharge, every active potential is going to have a release of neurotransmitter at the end. Eventually, you're going to run out by doing too much. So you run out. And that produces fatigue. Progressive inactivation of postsynaptic membrane receptors. The neurotransmitter has to bind to that postsynaptic membrane. If those receptors are used up, there's nothing else to bind to, no effect is going to happen, you get fatigue. Or you can get abnormal concentrations inside that postsynaptic cell that ruins the diffusion of it. So that's what causes fatigue of these neurons to happen. The other thing is pH. The acidity or alkalinity of our blood also affects our synaptic transmission. Alkalosis, which is what? Acid or base? Base can cause seizures. Acidosis, comas. Okay, so acidosis is going to decrease excitability. If you're not excitable, you become stuporous, coma, keep going, all the different stages of altered mental status. But alkalosis increases excitability. That can have you get seizures. Hypoxia, so low oxygen environments will also affect synaptic transmission. High requirement of oxygen for neural excitability. Cessation of blood flow to the CNS for 3 to 7 seconds. That's all it takes. Can make you go unconscious. Just 3 to 7 seconds of no blood to the brain, no oxygen to the brain. Hypoxia will make you pass out. Drug effects also have effect on neurons and their actions. Sedatives, anesthetics make depolarization more difficult. It's harder to stimulate, so therefore you are in a comatose state during surgery. You're not awake, you're not doing anything, therefore the surgeon can happen. You won't have any problems. Caffeine is here, neuronal excitability. For those of you who just started drinking caffeine, because of all those studying you have to do, you will be getting headaches very soon when you stop drinking it. That's normal. It's just a rebound reaction from not having the drug anymore. Just drink some caffeine, it'll go right away. Okay, we'll do a little bit of sensory and we'll stop. So, definitions for our sense. Yeah, you have a question? Oh, you're raising. Okay. So, how do we perceive our environment? How do we know what's going on? How do we know what's up, what's down? How are you hearing me? Are you able to see what's on the board? Stuff like that. The stimulus is the type of energy, vibration, pain, warmth. There's one sensation we don't sense for. Anyone know what we don't have a sensory for? We can't sense wetness. We have no sensory for wet. We know that we can feel cold or warm, or we can feel pressure on the skin, or the hair is moving, but we have no sensation for wetness. The receptors, okay, they detect the stimulus. We have photoreceptors in our eye to get the light converted into electrical signal, which our brain can then interpret as vision. So receptors for everything. And they are transducers in that they convert the stimulus energy into electrical signals. Our brains, we are wired for electricity only. So we need electrical signals to the brain that we can interpret, and we know that we're hearing things, or we're seeing things, or we're feeling things. The pathway is how it gets to the CNS. What path does it take to get there? What do all sensory neurons have to go through in the brain as part of the pathway? Thalamus. Okay, so all sensory are going to have some sort of word, thalamic, whatever. That means it's going to go through the thalamus. And finally, we have the interpretation that occurs in different parts of the CNS. Hearing is temporal. Your vision is your occipital lobe, and stuff like that. So different parts of the brain interpret different things. Why didn't it work? Just give it a second. I'll come back up. There we go. Okay. So we have to encode this stimulus in different attributes. So we talk about the modality. That's the type of stimulus. Vibratory sense, pain. When you guys, for PAs, when you get into more of your physical exam for the neuro, you're going to do pain sensations and all the dermatomes, light touch, vibratory sense, position sense. All these things you're going to learn how to do, all talking about how the body works. We know where we are. So that's the modality. The intensity is the amount of stimulus. Very loud noise, sharp stabbing pain. What's the intensity of the stimulus? The duration, how long is it lasting for? And finally, the location. Where in the body are you hurting? Can you point to an area that it hurts? So that's what we're trying to do with a stimulus. Where is it located, the type, the intensity, and how long did it last for? So with transduction, we need an adequate stimulus. Again, the stimulus is to get us above threshold so that we can have an action potential, so that the communication can get to wherever it's got to go. Generator potential. This is the trigger action potentials if threshold is exceeded. Large stimuli are encoded for high frequency. So frequency is the amount of times it's going to go. So it's called frequency coding. The number of receptors that respond to the stimulus is population coding. With nerves, there's crosstalk, where you feel one thing, you'll feel something else in another area. Visceral pain receptors, chest pain. Well, chest pain can also be esophageal pain, because the nerves all go in the same area, and you have pain in this area. It could be your heart, it could be your esophagus, or your stomach. Those are the ones that are going to refer to that area. Why? Because the pain receptors kind of have crosstalk, and it has a wide area for population coding. Multiple receptors are being stimulated. So pressing harder on an area of skin stimulates more receptors. So if you push on your skin, the harder you push, the more receptors are going to be triggered with the stimulus. Therefore, your brain's going to interpret a bigger area that's being indented on your skin. So the harder you press, the more receptors are sensed or occupied, and your brain picks that up. So if we look at this first one, generator potential fails to reach threshold. It wasn't strong enough to get you above threshold, so nothing happened. Then the generator potential exceeds the threshold, so we are now above threshold. We get a response. The bottom part is the response. And then increased action or potential frequency, so more, temporal summation in time, more being fired. We get a bigger response. So the more receptors are being triggered, the bigger the area is. One of the things we're going to learn about is 2-point discrimination. What parts of the body can sense that there are 2 points being touched versus 1 point? And you'll see different areas depending on what part of the body, like your fingers, like a couple of millimeter difference, you'll feel 2 points. But like on the back of your calf can get to like 15 to 20 millimeters or even more before you start feeling that there are 2 points touching your skin. So that's what population coding is. We're showing how dense these receptors are. Like if you ever get food stuck in your mouth and your teeth, it feels like it's a huge chunk and your tongue's going crazy to get it out, but you can sense that. Whereas some places are, you don't have very many pain receptors in the antecubital space, which makes sense. Now we can do blood draws from it. It doesn't hurt as much. If you do blood draw on your hand, it hurts more because you have more pain receptors there. So as you're putting that needle in, more pain receptors are being stimulated to cause more pain. You don't have that much in your AC area, So blood draws don't hurt as much in this area. Same thing with different parts of the body. Some heart have more pain than others and so forth. Adaptation to sensory receptors. We don't want these sensations to go on for a long period of time. So some things we want to be constant, like pain. Pain you don't want to go away. You want to know that you're in pain so that you can move away from something that's causing the pain, remove whatever object's causing the pain, or if it's like an internal pain, hey, something's going on, something's wrong, I need to get something checked out. But other types of sensations you don't care about. Does anyone here feel their clothes? No. So that receptor went away. You first put it on and you felt it, but then after like 10 minutes of wearing clothes, you don't feel like you're wearing clothes anymore because you got used to it. The sensation phased out. See the difference? So as we go through this, we have tonic responses. Constant train of action in response to sustained stimulus. Pain and balance. You want to know if you're off balance. You want to hurt yourself. So those sensations don't go away. Pain, balance. Those are your tonic responses. They don't diminish. No adaptation, constant. Phasic response. Action potential declines over time. Smell, touch, temperature. Okay, smell. Okay, if I put a spray of cologne in this area, eventually through diffusion it'll go to the other side, but after a while of you smelling it, you can become nose blind, right? And you don't smell things anymore because you adapted to that smell and you don't notice it anymore. When you take a shower, okay, the temperature hasn't changed. You got used to it, so you turn the temperature up more to get hotter so you can feel better. Okay, your receptors diminish to the temperature. So you need to turn it hotter to have that change so you can feel the heat again. So receptors can be slowly adapting or rapidly adapting depending on the generator potential and the decay of it. So you can see here, slowly adapting, still has the grade stimulus, still has the stimulus there, and rapidly adapting, the stimulus generator drops away, and they still have the stimulus there. So we want to potentially ignore environmental stimuli that don't matter. Temperature, you get used to the heat or whatever the temperature of the room is, and then you're okay, and then you're fine. So it's adapting so we can forget about it. Pain and balance are the ones we don't want to forget. So sensations from skin and muscle, we have 4 cutaneous modalities that we can sense from the skin. Touch, vibration, pain, and temperature. Those are it. Those are the only 4 things that our skin can sense. Now, it picks up different sensations and feelings depending on what of those criteria are stronger than others. If pain and vibration are high versus others give you a different sensation. If temperature and pain are high, that's why if you get like really, really cold on you, it burns and it hurts, versus also very, very hot. Also burns and it hurts. Also includes proprioception. Proprioception is our position sense. How do I know we're upright? That's why like the little mobiles for babies are so important, because it teaches them orientation. They know the eyes are above the mouth. So that's why like, playing peekaboo is very important for babies, because it shows orientation, and that's how they know what's up and down. Sensory neurons travel to the central nervous system and peripheral nerves and our cranial nerves. The area of skin supplied by different nerve fibers in a single dorsal root. So I said before, what are the type of sensations that we do with the dorsal? Dorsal is always sensory, ventral is always motor. Okay, so dorsal root, and that gives us a dermatome. And these are our sensories. How do we know what's going on? So this is what a dermatome pattern looks like. So we know if you're coming in, let's say we have sciatica, okay? That is going to be pain in the red area, okay? And we know that that is S2. And if someone comes in with pain going down the back of their leg in this pattern, we can figure out that it's probably S2 that's causing the problem because of the dermatome that it's following. Make sense? So each spinal nerve that comes off goes to its own dermatome. And that's how our body is mapped out. The reason it looks weird at the bottom here is because if you think about evolution, we were created originally, foreign, whatever you want to call it, to be four-legged creatures on the ground. And then we stood up, but the dermatomes didn't change. So if you put someone in all fours on the ground, all the dermatomes line up properly. It's just like because now we're upright, it all switched. That's why it's all wonky in the legs. But if you go like a bear or a dog, it's all going to line up perfectly. So those are the dermatomes. So again, sciatica pain, we know what route it's going to go down, and depending on what thing. So in PAs, what you guys are going to do when you get to your nerve exam, you're going to do with your, like a pinprick, you're going to hit certain dermatomes, and you're going to say, let me know when you feel pain. And you just go. And if they don't feel pain in one area, then you can say, well, maybe they have a problem with C5, because when I pinprick in C5, the patient doesn't feel anything. So C5 must be the problem. That's why knowing your dermatomes are important and knowing the roots and where they go, because you're going to work backwards from that to figure out where the deficiency is. Touch sensation on our skin. We have Merkel's discs and Meissner's corpuscles. Just know what each one does. You don't have to go crazy with this. Just know what they do. Merkel's discs is steady pressure. Meissner's corpuscles, rapidly adapting for changes in skin context. When things change on your skin, sensations change. Your Meissner's corpuscles. Merkel's discs are for like steady pressure, so like pushing down on your skin, whatever. Ruffini's endings, these are for stretch, slowly adapting. So stretching of the skin is your Ruffini's edges, endings, which sense the stretch of your skin. The Pacinian corpuscle, these are for vibration, hair follicles, displacement of hair. But that displacement of hair allows you to feel like when I said when you go in the water, you know you're moving through the water because your hair follicles are moving with it as well. Any swimmers here? Okay, so what happens at the end of the year before the big meets? What do you do? And what happens when you first put your foot in the water? You can't feel anything. If you ever shave like your legs for swimming and put your foot in the water, you don't feel the water at first. You will in a second or two, because the hair follicles don't move anymore because they're all gone, because you shave them all off. It's just a weird sensation, but the only way you can feel that is when you actually shave your hair and then go in the water, you don't feel anything. So that's why hair follicles, they do have some movement, and that provides like wind or water. So here's that touch sensation I was talking about before, the 2-point discrimination. 2 points of contact I've perceived as 2 distinct stimuli. Okay, greatest spatial resolution, fingertips and your lips. So if you look here, first finger can detect 2 millimeters of difference between 2 points. You will sense that you have 2 points touching your fingertips. That's how sensitive your fingertips are. That's why we have you use your fingertips in the pads when you're feeling for like pulses, because they're very sensitive. The tip of your finger is not, but the pad is. So the pad of the finger is used for like, feeling for pulses or whatever you're doing in your physical exams, feeling for lumps and whatever. The pads help with that. And then you can see here, your calf is 48. Okay, so 48. So almost 5 centimeters before you start noticing that there are 2 points touching your calf. That's how wide it is. But you look at your fingers, it's like 2 millimeters. And just showing again, distribution of these sensors, the receptors. They're stronger in certain areas. You're going to pick up more things with that location versus others. Somatosensory cortex. So we have the whole brain mapped out. It's called the homunculus. And we know where everything comes from. So it's this little map called the homunculus. And basically, we look at the central sulcus. That central sulcus differentiates 2 parts of the brain. We have the pre and the post. Sensory is the post-central gyrus. All your sensory is sensed on the postcentral gyrus of your cerebral cortex. The precentral gyrus is motor. So this is the homunculus. So if you were to have an itchy nose, that receptor from that nose would go up through what relay in the brain? Thalamus. Then that impulse is going to go there, because that's the location of where in the brain we code for the nose, for sensory. Leg, trunk, foot. So if someone comes in with a stroke and they can't feel anything in their feet, I know the stroke was up here somewhere. If they're coming in and their face is down, they have a facial droop, head is right there. Oh, here's the face. So we know that area was affected because they have a facial droop. So I know where in the brain they had a problem. And then you know what blood supply goes to these areas. You can figure out what blood supply caused this issue to happen. So it's all mapped out. We know where everything is, and that's why I said before, when people come in with certain neurological complaints, you could figure out where the lesions are because we know this whole thing is mapped out. We know where things are. So that's the homunculus for somatosensory. Here's our temperature sensation. We have cold fibers and warm fibers. Now, I mentioned before, both types are rapidly adapting. So does that mean it's going to stick around for a while or it's going to go away? It's going to go away. Temperatures, it just goes away. Interesting thing about temperature. When you start going into the very cold or the very hot temperatures, it will cause pain as well. This is a safety mechanism. You don't want to go into like hypothermic environments and adapt to that cold and just sit there and die, essentially, from the cold. So by having the severe cold cause pain, you'll want to remove yourself from that environment to get away from the pain. The heat is the same thing. So you don't want to burn yourself. Because if you're burning yourself with fire, it's going to cause a lot of pain as well. The other reason why pain and temperature use the same neurons. So the same neuron that's going to sense pain in an area is also going to sense temperature. They go together. So that's why with PAs, when you go to your exam, you don't test for temperature, you test for pain. Because if the pain works, we know the temperature works. Because they do the same exact neuron. They follow this, it's the same wire. Does the same exact thing. So safety mechanism. Too cold or too hot will cause pain so that you remove yourself from that situation. In these temperatures, it's okay. You're not going to, it may be hot, but you're not going to die from that. Pain sensation, nociceptors are free nerve endings. So your paper cuts hit those nociceptors, cause a lot of pain. We have a lot of obviously we know we have a lot of sensory receptors in our fingertips. So that 2-point discrimination is only 2 millimeters. So there's a ton of nerves in your fingertips. That's why paper cuts hurt so much, because there's a lot of nerves right there you're cutting into. Nociceptors have high threshold and should be silent. That's your pain threshold. When do people start experiencing pain and they want to stop? Okay, some people have very high thresholds for pain. Some people very low. I will tell you that NBA players have very low thresholds. The minute they feel any type of like tiredness in their legs, they're like, "Can I stop?" I'm like, "No, you gotta keep going. You gotta keep go for your heart rate." I've also tested hockey players. Those guys don't stop. They will keep going until they die. So you have to tell them, "Okay, the test is over. You can stop now." They all want to go who had the most time. If they're on the bike the longest, they win. So they all go crazy. But again, pain thresholds. People have different pain thresholds. Superficial pain is our body surface pain. So you scratch yourself, that's superficial. You can really see where it hurts. Usually going to be sharp, highly localized, so you know exactly where it hurts. Point to an area, where does it hurt? Has myelinated large fibers. We also have delayed, diffuse burning sensation that lasts longer than the initial pain. And these are delayed due to unmyelinated fibers. Unmyelinated means it doesn't have the myelin on it. There's no resistance. There's no insulation on the wire. It's not going to go very fast. Myelinated has that sheath. They go fast. So delayed pain. Someone punches you really hard. At first, it's going to hurt because you got punched. And then a little bit later on, that other wave starts happening. That's that delayed pain. So that's what that is referring to. Deep pain is going to be in your muscles and your joints. So if you ever start working out, your joints are going to be screaming the next couple of days. That's deep pain. Again, biologically necessary that they are slowly adapting so that we know that we have pain, so we can either get away from the pain or go seek help for that pain. So NSAIDs, non-steroidal anti-inflammatory drugs. Why are they pain desensitizers? They block the production of these inflammatory pain things like prostaglandins and leukotrienes. These cause pain to happen. So when you take an NSAID, ibuprofen, Advil, Motrin, they block the production of the pain-producing hormones or chemicals so you don't have the sensation of pain. They're not going to fix anything. They just make it so you don't feel the pain. Muscle relaxers. They don't do anything for your muscles. They just make you tired so you don't use your muscles so you can heal. That's it. They don't do anything for your muscle. You're tired. You don't want to move. You rest and your muscle get better. So pain sensation, visceral pain. Pain arising from the internal organs. This is usually poorly localized. So like belly pain. Well, where does it hurt? Oh, my whole belly hurts. I don't know where, but if they say, oh, it hurts right here, then I know it's something superficial. That's why with like cardiac pain, someone comes with chest pain, I'm like, where does it hurt? Like, oh, it hurts right here. I'm not thinking cardiac at that point because they pointed to an area on their chest where that pain hurt. True cardiac pain is going to be this deep visceral pain. They're going to say, oh, it kind of hurts over this area is where it hurts. That's what I'm talking about with like delayed referred deep pain is because of what's going on. It's the organs that are having the problem. And this is the crosstalk. So we have skin nerve fibers, visceral nerve fibers. You see how they all come in together there? That's where the crosstalk happens. And that's why you get like GERD pain seeming like it's heart pain. Because it's all that one area. They all kind of get some little crosstalk going on there. And then here's like another thing I told you before. This is the overlap of this type of pain presentation. So here's your esophagus and your heart. You see how they're overlapping it a lot? Esophagus referred pain feels like cardiac pain. It's all in the same area. Appendix, small intestine. Where's your appendix in this body? It's right here. Where are they going to feel it if they have appendicitis? In the center. Why? Because the way the crosstalk works with the nerves, that's where they are. So when you guys are PAs, you start doing your abdominal exam, you're going to see, well, for appendicitis, where are they going to start having the pain? You'll figure all that out. And we're done. We'll do this tomorrow or next week. Okay? Have a great weekend, everyone. Have fun tomorrow.